family mode.
Hi everyone, I'm back with my friend Philip Norris MD, and we are going to continue talking about coronavirus. He is, now correct me if I've got it wrong, an immunologist with the second largest blood bank in America or the world?
Oh, I don't know. There may be, I don't know about the world. I would just say U.S.
It's a pretty big
It's pretty big. The U.S. does transfuse a lot of blood, so.
I want to start with, first our topic today is when people talk to us and say, oh, COVID-19, it's just like the flu. I don't think it is, and you can tell us more about that. But there was a story that came out yesterday that the World Health Organization said that the asymptomatic spread of coronavirus is very rare. And there was another story earlier in the week that it was one of the, maybe it was the New England Journal of Health that had to retract 2 studies that made a major claim about coronavirus. And it's bad enough as it is that we don't know what's going on, and we're looking to our leadership, depending on who you talk with, whether or not this is reliable leadership in the CDC and the WHO and others that are supposed to be protecting population health. And we don't know what to believe anymore. So, Philip, help us out.
Yeah, I'll tell you what I think. So I liked your title, made me think a little bit, and I did a little bit of reading and talking and trying to figure out, so what is this compared to the flu? So that's one of the things we want to know early on. Mortality for the flu is probably less than 0.1%, slightly less than that. So a little less than 1 in 1,000 people infected with influenza die. I'm going to focus on death since that's a sort of a solid endpoint. You don't want it. So we have a reasonable handle on how many people are dying of coronavirus. Now, I think we probably missed some. Someone dies at home suddenly, and we don't know that they don't get tested. In the tragic case of Floyd George, it turns out he was actually infected with coronavirus. That doesn't look like what he died of, but so we know that people have it who are dying that we're missing, but we have a reasonable idea. If you go to the hospital, you get tested, you're positive, and then die, that you died of coronavirus. So we have a much less reasonable idea of how many people got infected because we haven't done wide-scale antibody testing. That's just getting ramped up now. And I think we're getting our first peaks at having the numerator, the top of the fraction, which is how many people die. The denominator, we're starting to get our hands around that. So how many people actually got infected? So I'll tell about 2 stories. One, there was the Diamond Princess cruise ship. I don't know if people remember that one. It got stuck in San Francisco. I think it was in our bay here. They wouldn't let them offload, so they went down to Miramar Air Force Base down in Southern California. And Lyle Peterson is one of the guys at the CDC. He's the head of the arbovirus branch, which is the mosquito and tick-borne diseases in Fort Collins, Colorado. He flew out to really do solid epidemiology on that. So they really were able to test everybody on the ship, whether they were infected or not, and whether they died or not. And on that ship, it was an older crowd on the cruise ship. So they had, I can't remember the exact numbers. It was something like 1.3% of people who got infected died. So we're looking at, that's more than 10 times as bad as influenza. Now, if you age adjust it, maybe that, if you try to extrapolate that out to the population that's a little younger, maybe it'd be 0.5%, which is still maybe 5 times as bad as the flu, maybe 8 times as bad. So we're in this close to 10 times as bad. So the other area where we're getting some data, New York City. There were a ton of infections in New York City. We know that from the news, right? So over 17,000 people in New York City died. That's a lot. How many people got infected? Well, there are something like 8.3 million people in New York City. And we're getting some data out. The New York State Lab has been doing some antibody testing. And we're seeing maybe some neighborhoods had 40% of people got infected in the crowded, poorer neighborhoods, mostly minority neighborhoods, we saw up to 40%, according to the New York State lab figures. But overall, maybe it was 14%, maybe a little higher. So we're looking at blood bank donors. We saw about 1% in March, 10% in April, 8 or 9%, I guess, in April, and we expect that to be a little bit higher. So, and blood donors tend to be a little wealthier, tilted toward Caucasians or the people who donate blood. So it's not totally representative of the population. So the number in New York of people who got infected, I'd say the upper limit probably is about 20%. So if you think 20% of 8.3 million, that's about 1.7 million. So if you divide 17,000 deaths by 1.7 million people infected, that gives you a 1% death rate again. So I think we're settling in around this maybe 1% death rate number, which is 10 times as bad as the flu. If you look at SARS-1, the first SARS virus that hit in 2003, or 2002, 2003, that death rate was closer to 10%. So I would say this virus, it's not the flu, it's not SARS-1, it's in between. So, you know, you have a 100-fold difference between influenza and SARS, and SARS-CoV-2 looks like it's about in the middle, 10-fold worse than the flu. So you don't want it. So that takes us to what do we do about it and how do we tackle this as a nation? So on the West Coast here, we've got a lot fewer infections. I think it's a lot due to population density, rapid transit. You know, we're not packed like sardines on a subway. So we had, we also socially distanced a little bit earlier, maybe in terms of when the wave got to California and the West Coast. But really, I think it's population density that's driving our lower infection rate. So there's more complacency here. I was up in Sacramento.
I think of San Francisco and major U.S. cities as densely populated. You're saying something that's opposite of what I'm expecting.
Oh, okay. Well, so I live in San Francisco now. I went to medical school in New York and used to live there, and actually got recruited back for a job back there maybe 5 years ago. It's incredible how much more dense New York City is if you're in Manhattan compared to
not anything in the U.S. compared to New York, but that's not.
Yeah, San Francisco is a low-rise, low-density city, really, in reality.
Okay, I'll take.
You know, there are areas where it's a little more packed, and you know, our hotspots up here are the poorer neighborhoods where people are more densely populated. And LA is more densely populated than San Francisco, and it's having most of the cases in California. So, you know, one of the things I did, I went up and saw my folks in Sacramento last weekend, and here everybody wears a mask. Out in the store, up there, I'd say only 20% of people were wearing masks in stores. It was mostly the elderly. So that was surprising to me. So I do think, you know, just to kind of wrap up this part of the introduction of today's hour, we're finding the mortality is probably a little less than 1%, but close to that 1% mark. So it's bad. It's also pretty transmissible. So I suspect we're going to see more aerosol transmission than we originally thought, and less on surfaces. And you can still get it from a surface, especially a hard surface that's not in the sunlight.
And that was debunked this week. I don't know what to believe.
Well, I think, you know, that's the problem, is we don't know a lot. So this is a brand new virus. It's been with us for, what, 6 months? So the fact that we don't know a lot of things is not surprising. And we just have to react as new data come in. I do think we're learning and we're doing better, but it's still deadly. So even though it's percolating along at a low rate, it still has a high mortality.
What do you make of yesterday's WHO commentary
about the asymptomatic transmission? Yeah, so that's you get into modeling, and modeling is as good as the data that you put into the model. I still don't think we have great data on how this spreads. So the modes of spread probably are, you know, people with respiratory secretions, either droplet or aerosol. So droplets are bigger and they fall faster. That's really the difference between droplet and aerosol. And aerosol are little small droplets that hang in the air for up to 12 hours because they're neutrally buoyant. They're so small. The question is with an aerosol transmission, so there are some viruses that have great aerosol transmission. So I think the star virus in terms of that kind of transmission is measles. If you're not vaccinated against measles, even if you are vaccinated, you can still get infected, but you're much less likely to. But before we had a vaccine, you know, measles ripped through communities, and they can map, you know, who was infected and who got, and to whom did it spread. And with measles, you know, if somebody walks down the hallway by my office door is open, and they're just breathing, I can get measles. It's that infectious. So that's the high end of transmissibility. This virus looks like it's a little more than flu. Flu is pretty much droplets. This one looks like there could be enough virus. So the reason an aerosol, the very, very small particles, might not be infectious is if the virus load isn't high enough in your upper airways. So if you have a ton of virus in your nose and your mouth, then even the tiniest particles have enough virus in them to infect a new person if they hit that person. So the question is how much virus is in the nasal passages? And for this virus, it looks like we may have super spreaders. So some people have extremely high viral loads in their upper airway passages, and those kind of people could potentially spread by aerosol, whereas other people, you know, 4 out of 5 probably have somewhat lower virus loads in their upper airways, and they would spread by the usual droplet, and they may not infect that many people. So it may be a few, you know, every 5th person is a big spreader, but most people aren't. That's probably what's going on, but we don't have enough data to support that. In terms of asymptomatic, I think that they're just monitoring. The way you define whether an asymptomatic person can spread it is you look at people who do get infected and when were they exposed to the person who infected them. Were they exposed just in the pre-symptomatic phase or asymptomatic phase, or was it after the symptoms developed? And that's how you would work out, was it pre-symptomatic transmission or not. They've certainly done studies in China of people who had no symptoms and had plenty of virus in their upper passages. So it's certainly possible. The degree of spread, which is due to asymptomatic versus not, it looks like maybe the WHO has more data. I haven't read anything in detail about it that suggests that mostly it's symptomatic people spreading it. You want to talk about the hydroxychloroquine trial?
You're welcome to talk about that. I was going to ask you more flu versus COVID-19.
Oh, let's keep on the flu thing. We have plenty of time.
I think you're going to tell me we don't have enough data yet for all of these questions, but they're the ones that seem to keep coming up. So I'd rather have the immunologist tell us, I don't know, than me saying, I don't know what I read. Like the flu, can you catch COVID-19 more than once? And is it the same, or is it mutating and we're likely looking at different strains?
Yeah, so we don't know about COVID-19 in particular. There are these stories of some people who stay RNA positive in their nasal passages for a long time. So it's possible there could be persistent infection. I have heard that they've, with influenza, a surprising study, which I haven't read myself, but I've heard about, where they looked in the lungs of kids with flu and found it a few months later that it was still there in a few cells, maybe even replicating. So, you know, it's possible that these viruses cause a persistent infection in some people. There's a longer tail of infected cells than we realized. So that's something I think we really need to know more about. Why do some people shed virus for so long? And some people go negative on the PCR and then they're positive later. And that could be they're just at the threshold of detection. I think this crowd is, you know, the medical device crowd is probably pretty savvy about understanding that you could have something that's near the cutoff that would be negative one day and positive the next, and that's just sort of assay performance. And then this reinfection question, there was a study done with seasonal coronavirus back in the 70s, I think in the 1970s, where they intentionally infected 15 people. And of the 15, I think 10 of them, they were able to get a productive infection in these people with the usual coronavirus that circulates. I think it was 229E. So that there are 4 strains that circulate around that we all get infected with. And the other 5, the 5 who did not get infected, had higher antibody levels against this seasonal coronavirus. And then they measured the evolution of antibody responses over time in these people and their symptoms. But what was interesting about this study is it came back a year later with the exact same virus. Coronaviruses don't change a lot like other viruses. And they were able to infect all 5 of the people who were resistant the first year because their antibody levels had dropped down over time. So they all got infected, and one of them had symptoms, but most of them had very mild disease, no symptoms. And then of the 10 who were infected the first year, I think they were able to reinfect 6 of them or something. So the point of this long story is that with seasonal coronaviruses, it seems like we get reinfected with the exact same virus every year or two, and it just circulates around, and usually we don't know it. It's asymptomatic, but we're carrying it, and we could spread it to someone who's never seen it before. So I expect that would happen with this virus if it goes on to behave like that. And what we hope to see, and we don't know, is that a second infection with this would be mild. And the corollary is we hope to see that if a vaccine induces a good immune response, that infection after vaccination would be mild. So we don't think we'll get a sterilizing vaccine. I don't think we will. So I think we'll get a vaccine that protects us from disease but not infection, is what I think we'll see.
That's interesting. Could you expound just a bit about that last sentence?
Yeah, so when we talk about protection with vaccines, so people say the flu vaccine doesn't work this year, it's only 30% effective. We might hear that in the news, right? Because it wasn't a great match. Well, that's true in that it prevents maybe 30% of infections, but it does prevent death from influenza. So you're much less likely, if you get infected with flu, you're much less likely to die of influenza.
You said infection versus disease, not infection versus death.
Well, death is a manifestation of disease. So that's the far right end of the spectrum of disease. So disease is when you feel bad, right? So when you get infected with influenza or SARS, it's not really the virus that's making you feel bad. It's that your immune system is getting turned on, particularly your innate immune system. And these cells recognize danger signals that the viruses have, and they all have these shared characteristics. And so your immune cells secrete cytokines, which are proteins that go out in the blood, and they make you feel terrible. So one of them is TNF. It's called tumor necrosis factor. Its other name is cachectin because it takes away your appetite. Cachexia is when you're very thin. So it is the one that makes you have a fever. So these cytokines make your body temperature go up. They give you body aches. If I took cytokines and just injected them into your blood, you'd feel terrible. And in fact, interferon alpha was a treatment for hepatitis C before we got our new drugs that are curative. And a lot of people couldn't tolerate that treatment. The interferon helped the immune system kick the hepatitis C out of the body, but it made people feel terrible while they took it. They felt like they had the flu. So every week they'd go in and get their infusion and feel terrible. So the immune system, that's part of the disease manifestation. You feel bad. So if you could get infected with the virus, and we all have viruses in our bloodstream right now. There are tortenoviruses. We have a whole virome in our body that's going around and not bothering us at all. So they're what we call commensal viruses that are just part of our biosphere that we live with. So you can have an infection but not be sick with it. Does that make sense?
It does. I'm going to show you a screen. This is from, let's say, a month ago.
Okay.
And I have both points of view on the phone because I know a few people pretty well who are on the line. I asked this question. I was asking about, phrase it perfectly, but I asked, do you believe reopening the economy now will result in unnecessary deaths? What I meant is more than otherwise would be the case if we didn't. And 56% said yes, opening too early will have a positive correlation with people dying. And the remainder said, let's do it anyhow. Because either A, I believe it will, but the cost benefit it's worth it. And 1 in 4 said, I don't think it has anything to do with it. So what are we waiting for? How would you have answered, and what would you tell the various factions here about their answers? We had 301 votes.
Yeah, so I think, I'll be like Clinton here. What do you mean by reopening? So I do think we have to somehow as a society adapt to this. We can't just say everybody stay home. It's fine for people like me who have a desk job and I can work, and I'm working more than ever. So I feel very lucky, right? But there are aspects of our economy that you just can't do that. In fact, in our own lab, we have people working away here. But I think we have to have a new normal. So when I was in Sacramento and people weren't wearing face masks in the store, I was shocked. So I do think that we have to have new rules of society to keep this at a low roar. I think that we will have, this 1% mortality rate is probably going to stick. Maybe it'll be slightly lower if we're in a situation where we've flattened the curve and we're not overwhelming the healthcare system. But every infection is going to be dangerous, so we want to avoid it. So with reopening, there are some things that you need and some that you'd like. So bars, rock concerts, sports venues, I think that may need to wait till we have a vaccine or therapeutic that works, because I do think mass gatherings, you just need one super spreader and you have tons of infections. Another way to reopen those mass gatherings would be if we gave up some freedom and allowed contact tracing, where our phones would tell us if we were next to a COVID person and we needed to isolate for a week after that contact. So we could do that. I'm not sure our society will be able to do that.
I have a friend in Italy who was swearing out anyone who would read it on Facebook, that there is an app being used in Italy called Illumi, I think, which is basically a contact tracer, and was excoriating anyone who would be so selfish as to not install it and be, socially responsible. I know.
Yeah, I'm going up to Canada next week, and I think they're going to put an app on my phone when I get there, the government, and track me for 2 weeks to make sure I stay where I am. And if I'm not, I'll get a visit from the police. So
how does that work? You're going to be stopped at the border. They're going to take your phone and install an app?
That's what I've heard. Yeah, from people who've done this, who've gone up there already.
Long wait.
It's the deal you make. You want to go into the country,
How are you going into the country? You're not Canadian. I thought the border was still closed.
It is. I'm a dual citizen, so we have a house up there. So we're
look at me. I'm an immunologist. I have 2 houses. I get it.
I'm married a Canadian very well.
Okay.
So
I think your answer was
giving up some freedom. If we want full opening, we need to give up some freedom. But what you could do more practically, so people who are looking at their own companies, we've reopened our lab mostly. All our desk jobs are still at home. I mean, I'm an exception, I guess, because I come in a couple of times a week to meet with people, but really, I don't need to be here that much. The people who do need to be here are the people at the bench, and we've done scheduling so that people are not overlapping as much. We've reorganized the labs to decrease the high density areas, and that may be as simple as moving one piece of equipment out of a given room into another room if it's in the workflow, so that you don't have 2 people working simultaneously side by side, but you can spread them out. So we've done a lot of that kind of engineering in the workplace, and I think that's something for this crowd on the call that we would want to do, to reopen your workplaces. And I do think we need to reopen now. I think it's safe-ish to do that, and we need to move forward.
I'm also going to share, before I relinquish my screen, this is where I go for the latest information on the virus. Ometers.info. And I'm fascinated by this column, deaths per population. So I'm going to sort by population. largest population, China, India, US, Indonesia, and you look at the deaths per million people, 342, 7, 10, 176. And I threw a question up on LinkedIn asking about this, and I'd much prefer your theory about how these are exponentially different among the most densely populated countries in the world.
Yeah, really good question. So, you know, I don't know, but I've heard that there may have been some underreporting in China.
Fair enough.
I know scientists there who are fantastic, but the government is a different story. So I'm not sure I trust those numbers entirely.
Could be a difference of 100-fold. It could be if you just want to doubt every information that comes out. But would you say the same of India, Indonesia, Pakistan? I mean,
yeah, there I think we do have pretty good public health here, where we have pretty good mechanisms of reporting, and we're able to capture the deaths. I think that if you're not capturing the cases and understanding why people died, that could be a reason why you might see a lower rate in a place that's has less resources. So we're seeing very little in Africa in terms of cases even, and I think, you know, there no news may be bad news, because if you're not detecting it, you may not be understanding the magnitude of the problem. So I think part of it is reporting. I think that might easily be most of the differences that you're seeing. But the USA definitely had a massive outbreak in New York with high mortality, and partly maybe because the systems were overwhelmed, but I also think we're better at counting cases probably in terms of the number of deaths.
Sarah reports that 30,000 people have died in New York. Joanne asks, is it true that doctors are being asked to report non-COVID cases as COVID related? I don't know where she would have heard that, but have you heard anything to that effect?
You know, this is where we get into politics a little bit. So how you report cases, is it likely COVID or not COVID, especially when testing was difficult in the beginning. So how we do influenza death reporting is we lump it together with pneumonia, because not everybody gets an influenza test. So the CDC actually has their death rate from influenza is an estimate based on influenza or influenza-like illness.
I've been compelled as a health service announcement that while my eye was itchy and I rubbed it, I haven't left the house in a week.
I love it.
I'm okay touching my face.
I think you'll be okay.
Okay.
But yeah, it is hard not to touch the face.
Especially with goatee, I find that I'm doing this all the time now. So it's ironic that I grew a COVID goatee.
That's
only
right.
I touch my face more.
It's your COVID catcher.
Yeah.
Yeah, it's not good for an N95 either.
Oh, I definitely want to talk to you about masks as well. Victoria asks, okay, what's the best material for a mask?
Okay, well, let's finish this little public reporting of cases and how it works. So, you know, I think in Colorado there may have been some controversy about how they were reporting the cases, and so I think both sides are saying the other one is undercounting and the one is overcounting, because there's a political movement to deny the severity of this disease, not just in this country but in other countries. You know, in Brazil, the president said to stop counting cases centrally and stop posting it because it didn't look good. They actually have more deaths per day than we do now. So I think that gets into the politics, which is unfortunate. I don't think this should be a political issue. I think we should come together and do this in a rational fashion, but that's my opinion. So you want to talk about masks?
Yes, please.
Okay, so there are the surgical masks. I've got one here. These protect other people from you. They leak air a bit, but basically they're snot catchers, right? So they get your respiratory secretions as you speak and block most of it. Some can still get around, but they're pretty good. So this protects other people from you. The N95, or what we can get the KN95, which is just the Chinese standard. So I looked up the difference between N95 and KN95. They're pretty similar standards. The KN95s are made in China. They're the Chinese standard for N95 that filter out, I can't remember the exact numbers of particles at a certain size. I think it's down to 0.3 microns or something like that, 99.5%. But anyway, they protect you. So they have a filter that filters out the droplet size that would carry virus particles. Now, it doesn't go down to the size of a virus, but you know, it's unlikely a naked virus is going to be floating around in the air. They're going to be in a droplet. So they're pretty good, and they're what we use in the hospital. There is one level higher, which is a positive airway pressure respirator, and that's an actual hood that goes over that has a battery pack fan. There's a battery pack on the belt, and there's a fan that blows air down, and it sucks in air through a HEPA filter. And the HEPA filter is a higher level of filtering, and so that is the safest way, and of course that's the most cumbersome. That's what we use when we see patients who have active COVID. That's what we use in our BSL-3 lab when we're culturing the virus. So I have SARS-CoV-2 is arriving today from the government repository, and we're going to work with it in the lab using these positive airway pressure respirators. So those are sort of the three level. Oh, and then the other is the cloth mask, which again is like the surgical mask. It protects you. One thing I like, this is a Gaiter, neck Gaiter. I don't know if people have seen these. There we go.
Okay.
So it just slides down over the head and sits around the neck, and it's easy to go up and down. Mm-hmm. So I wear these when exercising. So this is good. So I bicycle to work and use my neck Gaiter. So those are the sort of different levels of mask. But think surgical or cloth mask, you're protecting others from you. And if they're not wearing one, they're disrespecting you and not protecting them. So one of the things we had to do at work here is really change the culture that unless you're in an office alone, you need to wear a mask. So anytime I go out in the hallway or have a meeting, even though we're 6 feet apart, we still wear our masks. So that's a big cultural change. And I think we need to keep that going until we get a vaccine.
And that's your mask and not your neck Gaiter.
You know, in the office, I think this is a little easier to wear. The neck Gaiter, for long periods, it covers up a lot of the face. It gets kind of near the eyes. So I find the surgical mask just a little easier to wear on a day-to-day. If I'm wearing it for longer periods.
Out how to make a comfortable nose and mouth covering.
Yeah, so that's important. And then, you know, we're flying to Canada. We're all going to wear KN95 masks for the flight because that's somewhere where you're in an air supply with someone who's nearby and you can't get away. So if you can't socially distance, then I think an N95 is better to protect you because that protects not only people around you, but from you, but also protects you from people around you.
Fred asks, does the flu have long-term complications, such as lingering organ damage, etc., reported with COVID-19, not to mention the rare children's Kawasaki-like side effects? And thanks for 2 excellent presentations. They love you, Philip.
Oh, you're very kind. Thanks.
It wasn't me.
So let's start with the long-term complications. So influenza, you know, we have 30 to 50,000 people die a year of influenza in the U.S. The doctors in the old days used to call influenza the old man's friend because people who are elderly or sick tend to die from influenza. Also, the very young, if you're under 2 years old, they have higher mortality. So we call it a U-shaped mortality curve. What was interesting about the 1918 influenza was it was a W-shaped mortality curve. So people in that young age group, like the 20s, also had high mortality, which was different from other influenzas. So influenza can affect other organs. It can affect the heart, the brain. It can, in rare cases, severe cases, it can do that. It can get in the bloodstream. My lab did a study back in 2009 when we had the H1N1, and we looked to see if blood donors had influenza in their bloodstream. And using sensitive testing technology, we found that really they don't. So if you're asymptomatic and you're donating blood, even if you have influenza at the time and don't know it, there's not viremia. The only time we see virus in the blood with influenza is in the really sick, hospitalized people. And they can have long-term organ damage, just like the SARS-CoV-2. So then getting to SARS-CoV-2, why does it have so much organ damage? It's interesting. I think we're finding that it can get into a lot more cells than we originally thought. I've heard of some neurological diseases. The other thing about SARS-CoV-2 is that it causes a clotting diathesis is what we say in medicine. People tend to form blood clots. So they've looked in lungs, and there are some very big blood clots in the big vessels, which are bad. But they've also found what we call microthrombi. Thrombus is just a fancy word for a blood clot. So, and micro means small. So we find these little small blood clots in a lot of different organs. So that also could explain part of the reason why we're seeing tissue beyond just the lungs, because the lungs are the main site of replication, we think, and the upper airways. But it seems like, and through mechanisms we don't yet know, that this disease also causes blood clots that could affect a number of different organs.
I'm going to open the mic for Jessica, who has a question for you.
Sure.
So she is presently self-muted. If you could unmute yourself, Jess. No. Okay, then I'm going to read somewhat of a longer question. If the virus's cytokine storm depletes and exhausts the immune system, and then leaves the individual vulnerable to infections and possibly mortality from other diseases with little or no defenses, wouldn't that account for much of the confusion surrounding deaths from other diseases as being reported as COVID-related?
Yeah, really good question. So going back to our friend influenza, which we've studied a lot more than this, we do know that with influenza, there's the acute viral pneumonia that happens. But then a few days later, a lot of people, the classic story is they get a staphylococcus bacterial pneumonia after the viral pneumonia is kind of clearing up. And that is what kills the person, is they get a bacterial superinfection. And there is, we think, maybe it's a little bit of the lung damage, but we also think there's an immune change that goes on, particularly in the lungs of people after influenza. So it wouldn't be surprising if we saw that with SARS-CoV-2. I have not heard that superinfection with other agents is common in SARS-CoV-2. And in fact, they have looked, and particularly the data out of China. So they've looked to see if there are other organisms that they isolate. And they found that there is co-infection with other viruses, but it doesn't look like the immune system is predisposed to getting bacterial infection after SARS-CoV-2 or during that disease course. One thing that is interesting is that, so the immune system has the innate immune cells that I talked about earlier, also has the adaptive immune system, which is B cells and T cells. The B cells make antibodies, the T cells kill virus infected cells. And for some reason, the T cells go very low in SARS-CoV-2 infection. We don't know if the virus is causing these cells to die off. It looks like it can get into the cell, and maybe it triggers the cell to commit suicide once it gets infected. We think that may be what's going on. That's one of the things we want to look at in our research studies. But there is a phenomenon where the T cells go very low, and this happens in a lot of virus infections, that the T cell levels really drop during the acute phase of infection. So your question, could the immune system be compromised and allow other infections, is a very good one, and the answer is yes, it could, but we haven't seen a lot of that in our clinical data so far.
Michael has a really interesting question. How transmissible is the virus through HVAC systems?
Excellent question.
Ventilation, air conditioning.
Yeah. So from what I've seen, the big hotspots have been areas that don't have great ventilation. So there was a description in I think it was a Chinese hospital, that a lot of the transmissions came in a changing room. So healthcare workers obviously are on the front line for infection and do get infected and die. So there were some infected healthcare workers, and they shared a locker room that had very poor ventilation. And that was one of the hot spots of transmission. So it wasn't that it was being piped around the building. So situations where ventilation spreads infection for viruses are situations where it's aerosolized. So our friend the measles, that can get into a ventilation system and infect people a ways away. SARS-1, there was some transmission, we think, via the ventilation system. So in Hong Kong, there were I can't remember, something Gardens was the hotel, and there's a infectious virus in the stool in SARS-1. There is RNA we detect in the stool in SARS-CoV-2. We don't think it's infectious because nobody's been able to culture virus out of the stool. They can see the RNA in there, but they can't culture it out. But anyway, in SARS-1 you can. And a lot of people had modified their bathrooms in this condo in, I think it was Amory Gardens in Hong Kong, and they had improper plumbing where it was aerosolizing a little bit of every time the toilet flushed, and that got into the air circulation system, and the aerosolized poo went around the apartment building and infected a lot of people.
You said
so
poo.
It is possible.
I know I was just 14 there, but you said poo, and I thought it was funny.
You gotta get your son back in here. He'd love this stuff.
Yeah, I know. Really interesting questions coming in. I hardly know how to prioritize them. Anne asks, can you comment on how COVID affects patients with low IGs compared to flu? Is it more dangerous?
Yeah, so if you have,
I don't know what an IG is.
Yeah, I do, so I can help out with that. That was the first thing. I was going to back up the truck, and we'll talk about IGs a little. So IG is short for immunoglobulin, and those are the antibodies. So your B cells make antibodies, and that protects you from infection. And some people are born with low levels of immunoglobulin through immune defects. So common variable immune deficiency. There are a number of reasons why people may have. There's a low IgA, and so there are different flavors of these immunoglobulins. IgG is the one we think of in the blood that's protective. The first one to come up is called IgM. And then it's sort of the first batch. If you're making an assay, it would be your phase 1 or your prototype assay, and then the second generation would be IgG, where you make a higher affinity antibody, one that sticks better. And then there's one that's for mucosal surfaces that goes into your gut and into your airways and nasal passages, and that's IgA. So people can be deficient in IgA. So if you're deficient in any of these, you're more susceptible to some infections, and it's theoretically possible that they would be more susceptible to SARS-CoV-2. I don't think we have any data on immune compromised people and SARS-CoV-2 at this point, because it's just so new that we don't know the differential of how the immune system would react. And how the person would do after infection. There is some thought that if you had a less robust immune response, maybe you wouldn't get as sick, because a lot of the sickness that comes, if you look at when people get sick, they tend to get infected. They have maybe some cough or fever, but really the first phase is fever. They might have diarrhea even. Some of them have diarrhea to start. And then there's fever, and then the fever starts to resolve at about a week, and that's when they get sick. That's when they have trouble breathing, at about a week to 10 days in, and that's when people have to go to the hospital to get oxygen. And that coincides with when the antibodies come up. So one of the questions is, are the antibodies actually causing some of this problem? And that's one of the things we're going to look at in the lab is, what is it exactly about that immune response that goes wrong 7 to 10 days into the illness?
I opened the mic for Peter, but you said something that triggered a question. You talked about things that you're going to look at in the lab.
Yes.
Could you tell us what are the top 3 priorities of your lab right now?
Yeah, that's a good question. So we're at a blood bank, so we're doing a lot of work, and we as the Royal Weeks, we have a lot of people in the lab. It's not just me.
Are we talking hundreds of people, dozens, what?
We have about 70 people at the Research Institute, and I would say probably 50 of them are focused on COVID at this point. So dozens. And we collaborate with people all over the country and all over the world. So one of the big studies that we're doing is looking at the rate of antibody positivity in blood donors. So we're collecting blood from 25 centers around the country, and not just our blood bank. We're collaborating with all the big blood banks to look at how many people were infected. So we have a really good test. We put a lot of work into trying to validate the various tests that are available, because there are, as you know, hundreds. And so we're using one of the big manufacturers' tests, and we're testing all our blood donors for a few months, and then we'll test selected, 1,000 from each site from 25 sites.
What test to use? Which one is the best on the market? James asked, what's the best RT-PCR and POC-PCR tests? I don't know what those acronyms stand for, but you do.
Okay. We'll digress a little bit. So RT-PCR is reverse transcriptase PCR. So real-time PCR, sorry. And this is an RNA virus, so we do a reverse transcriptase. So I think there are a lot of good RT-PCR tests. It's not as hard a test to make. We're in the blood bank. We tend to work with Grifols, which is a company that makes blood bank testing, and it uses transcription-mediated amplification, which is an isothermal amplification. It's like PCR, where you can copy the DNA or the RNA. So we're using their test, and we're just getting the test batch reagents. So I don't know exactly how sensitive it is, but they tend to be in the 10 copies per ml or 1 copy per ml, somewhere in that range, 1 to 10 copies. So it's pretty good. And there are a lot of good tests out there for that. So I'm not so worried about the RT-PCR, and the POC is point of care. I know the Abbott had a problem with their swabs going into viral transport medium, that they had a sensitivity issue with their PCR test. They just now want you to stick the swab right in the machine. So I think that fixed that problem probably. For the antibody test, that's where the variability comes in, because it's a lot harder to make a protein, to express the protein in the right conformation that the antibodies will bind to it, and that you don't have background reactivity, cross-reactivity. So that's been really difficult. And there are a lot of rapid tests, and they just don't have the specificity that we'd want to see in a low prevalence setting. Most of your positives are going to be false positives. So we're using, in the blood bank, we're using the Ortho Vitros system, which is a chemiluminescent assay, has excellent specificity. We haven't seen any false positives yet, and good sensitivity too, and has a wide dynamic range. So that's what we're using in the blood bank setting. It's not, it's a big machine, so you would have to have a dedicated lab doing this. It's not a rapid test you would roll out for your own employees at your company or something.
I don't know why, but James wrote, "Oh, that's a volatile argument."
Oh, I don't know which one. I don't know which one was volatile.
Okay, James, I'm gonna give you the
elaborate, James.
Go ahead. Just don't go on too long. I know you.
Hi, how are you, Doc? Thank you so much for coming on.
Sure.
It's been incredibly informative, and you're awesome. Anyway, I digress. To the point of the sensitivity on the POC, IgM, IgG serology test kits.
Specificity. I don't have any problem with sensitivity. Specificity.
Well, so long as it meets the new federal standard, the FDA standards for specificity and the like, which is 90-95. What is your viewpoint on those test kits that do meet that FDA minimum and above?
I'll tell you a story. So the Abbott test, antibody test, it's detecting nucleoprotein antibodies. So it is 99.6% specific. That's pretty good, right? That's way above what the FDA requires. And we tested 1,000 samples in San Francisco. 4 were positive. Then we retested those 1,000 samples on the Ortho platform that has basically 100% specificity, and 3 of the 4 positives were false positives. We also tested with a neutralizing antibody test, and the 1 sample was positive on all 3 assays, and the 3 false positives were negative on the Ortho test and on our neutralizing antibody test we have in the lab. So that tells you that in a low prevalence setting, even a test that's 99.6% specific is going to give you mostly false positives in a city like San Francisco, where you're less than 1% at that time, less than 1% seroprevalence. We're probably up around 1% now. So if you're at 95%, which meets the FDA guidelines for specificity, and in New York City, even where you may have 20% people are positive, you have a ton of false positives. You would have, out of 100, you would have 5 false positives and 20 true positives. So but if you're in a setting where it's 1% prevalence, like most of the rest of the country, you would have 5 false positives for every 1 true positive that you have. So I don't think that the FDA, it depends how you're using the antibody test. If you're trying to do seroprevalence, you can, as a survey study, you can kind of work with that.
It's really, I guess, meant to be kind of a screen, right?
Yeah, I mean, people want to know if they're antibody positive, and so I think you have to have really high specificity in a low prevalence setting to use an antibody. So I'm not a fan of the rapid antibody tests for this situation. I mean, they have their uses, but I think for the current COVID situation, they're not as useful as we'd like them to be.
James, if I'm not mistaken, you represent someone that sells tests.
Yeah, I do.
What kind of test
Well, don't hire me as a consultant.
What kind of test do you sell, James?
It's the lateral flow rapid test.
Yeah.
So this is exactly what Doc is talking about here, and it's a very, very good test to do screening. And if it's a moderate in a moderate CLIA lab setting where it's doing a blood draw versus a pinprick, you have a lower prevalence of contaminating the sample, blah blah blah, and it lessens the potential for the false positives. So
Doc, you agree with everything you just said?
Well, so the folks at Stanford did one of these lateral flow assays, and they were widely criticized. It was put on bioRxiv, but I don't think it ever got published because it was the false positive rate. If you factor that in, the rate of positivity they saw was really not different from 0 if you look at the false positives. So I think even for serology, you need, unless you're in a setting like New York City where you know that there's a 20% positive rate, you can then tolerate some false positives and still get accurate data for a sero survey. Certainly not for returning results to people, I think, if it's not really specific. That's my opinion, but
you're zeroing in on the specificity, and
yep.
The test kits that we have are very high on the specificity side. So
yeah,
I get what Doc is talking about, and I agree. I don't disagree with him at all.
In the interest of time, Philip, if you're amenable, I'll give James your contact info, and maybe he can have a conversation.
Always happy to talk. And I do have to get off at 9:15 because I had another call.
So I have a comment from Peter, and then one I'm going to read out on Elena's behalf. Go ahead, Peter. I'm sorry I gave you that. There, you should be. Yep.
So I have a few things quickly. So first of all, Dr. Norris, thank you very much. You've been extraordinarily useful, and I agree with everything you've said. Your views align with mine. We should connect on Slack, please. Regarding the earlier comments you made about the transmission, Christian Drosten, the famous coronavirologist from Germany,
Yeah.
who has the Das Coronavirus podcast.
Yeah.
He believes that it's 50% transmission by droplets, 40% by aerosols, and 10% by surfaces, fomites.
I like that. That aligns with what I was thinking, and I'm glad to hear that matches Christian because he's a really smart guy.
Yeah, he's like, I consider him the greatest coronavirologist in the world. Second question I had was, do you believe or do you worry about flu season, regular human influenza, co-infecting patients that are infected with coronavirus and causing them to become super spreaders above and beyond the super spreaders that may exist just from high upper respiratory airway tract infection?
Wow, that's interesting. So at the beginning of the epidemic, we thought, if someone has an influenza positive swab, and we had very limited tests available for coronavirus, for SARS-CoV-2, if they're influenza swab positive, well, they don't have SARS-CoV-2, we're not going to see co-infection much. And then there was a nice paper that came out of China that showed that the rate of all these viruses was basically the same in people who did or did not have SARS-CoV-2. So I think, 12% of their hospitalized patients had positive influenza swabs, whether or not, and it was the same almost in those who were not positive. So we are going to see co-infections with these respiratory viruses, is the point of that. So your question, would the virus load be higher? I don't know that they actually looked at the SARS-CoV-2 virus load in the co-infected versus the not co-infected patients, but really interesting question that could be answered.
It's not so much that the virus load is higher. I worry about the sneezing and the coughing associated with influenza causing a patient that has upper respiratory tract coronavirus to become a super spreader.
Well, people cough plenty with the COVID. So I don't think I don't think that the influenza is going to make you cough or sneeze more necessarily.
The infectious people are people in their 20s and 30s who show no symptoms, but if they have the flu and they have high levels of upper respiratory tract infection, they will become a super spreader.
I see what you're saying. Yeah, so I think the super spreaders really they're those people who have, that Christian was talking about, who have a really high virus load. So it doesn't really matter how much you cough, it's really to do with when you're speaking or whatever, the little tiny droplets, are they loaded with virus? So I think the virus load is what drives people to be super spreaders, not their behavior of coughing versus not. But I think, are they more likely to spread the virus for a given viral load if they have influenza as well and they're asymptomatic? Well, that's certainly possible. I don't think that's going to be a main driver, though, of transmission. But it's an interesting theoretical possibility, and I think your thinking is good.
One final statement.
Okay,
one final. The worst things you can say to somebody is stay healthy because it sprays virus out of your mouth.
I love it.
I need to mute you, Peter, because I want to respect Philip's time. I also want to share very quickly my screen again. One of our members, Elena, asked the group, does anyone have extra de-identified patient samples laying around? I said, I don't understand. She said, we need nasal swabs collected from patients and preserved in viral transport media to comply with IRB guidelines. We need to purchase them from a supplier or obtain specimens. We need negatives to test our product with. Philip, do you have access to that kind of thing?
We don't have nasal, we're a blood organization, so we have a bunch of blood, but we've been, there's a site called BEI Resources. I don't know if Elena is familiar with that, but they've been putting together all sorts of reagents that are necessary. That's sort of their mission, BEI Resources. Okay. It started off in response to the anthrax attacks. It's a biodefense repository was the original name of it, and it's changed to BEI Resources. So Google them. Okay. They'll ship it for free. I mean, you have to pay $25 for the shipping, but they give you all the materials for free. It's a government-funded thing.
She'll be more than thrilled to hear that. You can see here that I have this button, invite people. I'm going to invite you. I don't know that you can handle the inflow.
I cannot be on the Slack channel. I'm a, I'm a busy guy.
Yeah, I suspect you are. Yakov says, how do we get in touch with Philip? I'll maybe eke out your email address to 2 or 3 people, but
Yeah.
I'm grateful, and I know on behalf of the group that they are as well. So once again, Philip Norris, thank you so much for your time and your talent, your information and education today.
Yeah, nice to meet you, Joe, and thanks to everybody for the questions.
Bye for now. Thanks, everyone.
Okay. Bye-bye.
Bye.